And here's a little synopsis also of how Immortality Institute members were involved a couple years ago. You did put out a call for soil samples in order to find bacteria that would have the enzymes in them already that could digest a lot of this junk. So a lot of people from around the world sent in soil samples. You then put the bacteria into culture and try to see if they would digest a lot of this junk that you're working on. And then you ended up, I know last year there were some reports that you did find find some enzymes, some bacteria that were able to digest some of the targets here, some of the junk between the cells, in the cells, this strand of SENS. And now, what point are you at this moment in the LysoSENS project?
So that's exactly right, Mind. First of all, I thank all our soil donors. Without your guys' submissions, this project would really have been impossible. And
yes,
I found lots of the stuff
much.
I found.
Yeah. Thank you so much to everyone. Yes.
Yeah, and I just want to assure people that I found lots of the stuff I found in soil samples that were submitted, although I can't usually acknowledge them individually because I have to combine some samples to achieve higher throughput. So I don't know who submitted something that did work, but many of them did work. And for some targets, the only ones that worked were submitted soil samples.
Oh,
so
okay.
Big thanks to everybody.
Sure, yeah, I know. Big thanks to everyone for all the donations. And you're at the point where now you do have some enzymes you've identified. What's next?
Yes, so there were three major targets that we had. The advanced glycation end products that accumulate in diabetes, the oxysterol, as you say, that accumulate in atherosclerosis, and a carotenoid substance called A2E that accumulates in macular degeneration. And so we've hit two of these three targets.
What about CML? What is that?
That's part of the advanced glycation end product.
Okay, yeah. AGEs. Okay.
Right,
gotcha.
AGE. So that AGE project is at an earlier stage. We have plenty of degraders of CML and other AGEs from the submitted soil samples, so microbes that degrade them, but we don't know yet any of the enzymes or genes that they use to do that. But for the oxysterols and for the A2E compound, we have enzymes now that are moving into cell models of disease. We've got some very good results from a cell model of macular degeneration that we didn't do. We submitted our enzyme to a macular degeneration researcher who was delighted at having an enzyme that degrades this bad compound that she has worked with for so long.
And I imagine, yeah, she was just absolutely ecstatic. So you are using these enzymes in cell cultures now. Is that
our collaborator is?
Oh,
yes.
Your collaborator. Okay, gotcha. Okay, continue.
So, and she managed to reduce the A2E content by about a third in her cell model with our enzymes, which is great. And so now she needs to look for pathology and things if that actually helps the cells to reduce the A2E.
Okay.
Yeah, and so we'll see what that does. And then it will hopefully move, some preclinical development route, next to mouse, and then companies will start getting interested and stuff.
Okay, yeah, so that's the next. I mean, I imagine, there's probably already a few companies that have their eye on some of these degraders. But the next step then would be mouse models after cell cultures, like in vivo.
Yes. So I have to say there is a big drug delivery problem because in cells you can get them to take up an enzyme that you just add, but to deliver that to an animal or a human is a lot harder, and you need to do some more engineering to get that to work.
Yeah. Okay. So that brings me to a question. It's a good thing that you mentioned that. Someone prior to the chat did ask a question. I don't know if he's here in the program tonight viewing, but. Lunar, he asked, how will these junk degraders be available for humans? Like you mentioned, hey, if just a cell culture, hey, it's easy to get enzymes in there. But with the drug delivery problems, is it going to require IV administration, or would someone be able to take a pill? Do you have any idea or any kind of, you can envision any process by why this will happen?
Well, yeah, so first of all, let me emphasize that, as you say, it's enzymes that need to be delivered. Microbes don't work.
No.
You know, the immune system either kills them quickly or they kill us quickly,
Yes.
so it's a bad idea to try that.
No, can't do that. You gotta have just the enzymes, right?
Yeah, right. So that's why we need to discover enzymes. That's why it's such a second big milestone to discover an enzyme in a microbe that's responsible for the breakdown. Yes, but to the question. So there is a precedent for having to deliver enzymes to human cells, and that's the heritable lysosome storage disorders. In these disorders, people lack an enzyme in their lysosomes. It's the same place where our junk accumulates.
Yeah.
Let me talk about the lysosome first, maybe. That's a little compartment.
Yeah.
It's a little compartment in a cell that's responsible for waste breakdown. So that's why it gives LysoSENS its name. It's derived from the lysosome.
A lysosome. It's like the garbage disposal of the cell, right?
Yes, exactly. That's the garbage disposal where stuff gets broken down, and also where stuff gets transported to for breakdown. And stuff gets always transported there, no matter whether or not the lysosome is actually able to break it down. For example, with these oxidized cholesterols in heart disease, the cells get rid of the cholesterols and push them into the lysosome, but the lysosome just fills up until you can see bloated lysosomes with that junk. It just accumulates because it can't break it down.
Can't break it down, okay.
And so what we need to do is get our enzymes into that lysosome, because that's where the junk accumulates. That's where they need to go.
Right.
And so heritable lysosome storage diseases in which a natural lysosomal enzyme is missing in a newborn. That happens, spontaneous mutations in the human genome that break such an enzyme. And these people die rapidly, sometimes by mid-age, sometimes early in age, from overfilling of the lysosome with junk that normal people can break down. And that was a big breakthrough to develop therapies for these people by just injecting the missing enzyme.
So it was an injection, basically. Yeah.
Yes, in that case it was an injection. And then it also has to get into the affected cells from the bloodstream. And that was really the hard part.
Oh, okay.
So that involves molecular targeting of certain cell surface receptors to tell the cells to take this thing up. You've probably, some readers are familiar with targeted cancer therapies where the drug binds to a specific cell receptor that's unique to the cancer cell. So it's the similar idea. It's a molecular tag that attaches to a target and goes into the lysosome. But the good news is, for the lysosomal storage diseases, which are heritable mutations, these enzyme replacement therapies in some cases achieved a complete cure, and in other cases achieved, substantial resolution of the symptoms. So in some diseases, people who would have died in their 20s are now approaching old age and are doing just fine thanks to these enzyme supplements. So we have a really, really good delivery system there that we just need to imitate.
Okay. So
and in some cases, for example, atherosclerosis, the same cell types are affected by heritable diseases that got cured as are affected in atherosclerosis. It's the macrophage. Gaucher disease is the name of a heritable lysosome storage disease that affects only the macrophages, where the cells are smart enough to give the junk to a cell type called the macrophage. And the macrophage then accumulates it.
Okay.
And that's the same cell that also accumulates the oxidized cholesterols in atherosclerosis. So it's really neat that we can just overtake that system for delivery.
Yeah, that is. I mean, it makes me feel quite positive about future LysoSENS type therapies, or at least these three strands of SENS that deal with the different types of junk.
Yeah, so for the other, it's easier because they are extracellular.
Yeah, extra.
So you don't have to trick a cell into. Yeah.
No, and in fact, with the Elan Corporation, with their antibody, I mean, they're using macrophages, aren't they, to attack amyloid beta?
Well, so it's not really well known how the antibody works, because the antibodies aren't supposed to cross the blood-brain barrier, and the thing still clears it out of the brain, so nobody really knows what the mechanism is by which this actually happens.
Oh, that is kind of odd. You're right. Yeah, I guess I read about that a little while back, and that did occur to me as well. It did seem kind of odd. Okay. So you're pretty optimistic, though, as far as things are continuing to move along here, and has everything gone about at the pace that you expected so far? Has it,
Well, at the pace that I expected, yes. At the pace that I desired, no.
no, right? Well, I know. I would love to see, faster adaptations, of course, or discoveries. We would all like to see it, but biology works pretty slow, right? It takes a while sometimes to get. Uh,
Well, at least at the scale that we are using, yeah.
yeah. I mean, you're really taking on something big here. Something.
Yeah, and we are a handful of students. That's all we are.